to see the phenotiphic changes. Alright? Alright, so that is the lecture 6. Alright, so I explained it. So if you have time, please watch it. So don't worry, lecture 6 is not continuous to lecture 7. So take your time until this weekend. Please have a read of it. Alright? Okay. So another thing is that some of you are starting to ask about assignment and test date, right? Alright. So usually for assignment, I think I'm going to do this week. I'll send you the assignment task this week. So basically, your assignment will be I need to write it down. So I tak write lagi. But your assignment, I'm going to give you to do one page. More like apa kita panggil macam poster pun, more into infographic, yes, more into infographic lah. You all suka kan? Why? You don't like infographic? Or you want a summary? Summary printer. Why you don't like infographic? I like. Because I'm not student. Oh, it's very hard, is it? Infographic? Okay. So in that case, if you want infographic, I'll change into a one-page summary. Okay? Summary of, okay, what I plan to do is that, okay, when you learn now about modification tools, so after today's lecture, you're going to learn about ZFM, RNAi, RNA interference, and then also you learn about talent, CRISPR. CRISPR I won't touch much. Why? Because there are 2 lectures with Dr. Tumita later that are going to make your head turn around with CRISPR. Alright? So I'll just touch what CRISPR, that's it lah. And what other tools used in CRISPR, that's it. Just a general vibe, alright? So, by knowing all these tools, find any research article, alright? Find any research article that use that tool. Either one. Whether it's the FM, whether it's talent, whether it's a tail, whether it's RNAi, or even cloning. You can even use a basic cloning paper. Alright? What are the genetic modification that were done in that paper? So summarize it in one page, not more than one page. Kau tak payah kecil sangat, sakit mata saya nanti. Alright? So in one page, make sure you have introduction of the research. Alright? Not the normal introduction or whatever. So the research that you choose, what does it aim to? Alright? So the aim of the research in your introduction, and then I won't explain more after this. I won't explain anymore. This is my last explaining. I'll just upload the instruction later. Alright? So if you want to ask now. Alright? So you explain the introduction based on your research paper. You read, okay, so this use talent. So the reason is that they want to optimize, so they want to cut a certain sequence in let's say a worms organism, something like that. So explain that in the introduction, and then methods. What sequence they are using? Alright, which part of the worm that they are using? This is an example lah. Alright, so the method that they use in order for that to genetic modification and the result. Meaning that what are the successful rates? So what are the outcome of the research? When they modify this thing, so what happened at the end? Alright? And then that's it. Conclusion boleh lah if you want to have some conclusion. And of course, the citation of the paper lah. That's important because I don't know what paper you will take. Alright? Summer paper, I tak syorkan. So how do you know sama ke tak? You bincang sesama kawan. I mean, kalau katakan group saya ada terpilih sama group sini, I know that you guys pun never discuss about it, something like that. So kalau tersama, never mind. Tapi try to find a current paper. Current paper tu maksudnya try to find 10 years from now lah, 2022, 2012, right? Try to find a research paper within that time frame. Where to search, tahu kan? Scopus or Web of Science, either one lah, alright? Google Scholar can make sure it's a good publication. Bukan pirates. What pirates? Try to find. Okay, now I want you to try to find research article in Scopus. Cari dekat Scopus. Alright? Pernah dengar Scopus before? Pernah kan? Okay. So try, because you are fourth year, I need to introduce you to a more scientific, bukanlah relevant, more apa convincing scientific page, alright, rather than a Google Scholar. Google Scholar is good as well, but it's quite vast. But in terms of the non-validated paper pun still there. Alright, so we want the validated one. So try to go for Scopus or Web of Science, okay? Alright, clear about that? Any question up until now? Yes? Uh-huh. Yes, can. Any tools for gene modification. This will also can. No problem. Okay, so for about tests, usually I'll done tests 2 weeks before mid-semester or 1 week before mid-semester. I'll ask you again, usually, what time is your suitable time. Sometimes students also request after mid-semester because a lot of lecturers do before mid-semester. So sometimes I tolerate it as well. Alright, we discuss it, don't worry. So I don't fix the date yet, but usually 1 or 2 weeks before mid-semester, or in special cases when you have something else to do, if majority said you want to do after mid-semester, I also okay. I don't mind. Okay? Alright, but bear in mind that after in the middle, I think starting next week, I'm going to teach another courses, so I'm going to be busy as well. Nila kan actually dia course sekali. But right? So tak apa, but we can kita boleh nego, jangan risau. Okay, negotiation is always open with me. Alright? Okay, so lecture 6, please have a look. Alright, for RLAI, so there's another tools there. So for these tools, alright, so such a recap because I'm not sure whether you will listen to lecture 6 or not. So but for lecture 6, we focus on two things, RLAI, and then for gene modification, we already explained it before. Alright, so today we're going to explore more about these three. Zinc nucleus, talent is mana I punya laser pointer. Tak mengajar seminggu dah. Tak ingat dah ni. So, okay, since it's a morning class, let me check whether or not kita boleh quit. Sebab kalau you dengar ada seorang, you ngantuk. So, let's try over some videos, okay? If possible. Kalau tak possible, biasalah, I jadi narrator. Tapi that's apa, dia punya sebelah mulia jadi school pagi. Kan? I tak boleh dapat long macam tu. Susah tu orang. Lagu Hindustan ya? Wow. Melawan ni. Mula bantuan plan. Okay. So basically okay. So have a look for a while. I tak tahulah macam mana I nak cuba. I tak nak kalah dengan orang sebelah. Kuat tak?
And those are basically an enzyme that can cut DNA at a specific nucleotide sequence. So in the one amino acid. So here it is ATG, and then the blue one is targeting ATC, and the purple GAG. And then these zinc fingers usually pair or work together in groups of 2 to 3, also bind to an enzyme called BOP1. And this you can imagine as like a pair of scissors. It's a DNA cleavage domain. And so if we have the groups of this sequence here, then we're able to make a cut right here by heterodimerizing these 2 cleavage enzymes. And so to remove an entire sequence like the one I've outlined in red, we need to make a cut here and another cut here. And that is why it's required that we have 4 of these groups of zinc fingers and BOP1 to do that. And that would be a minimum of 8 zinc fingers, or usually a maximum of 12, like I've done here. And so we can bring these zinc fingers into the cell by transfection, which is deliberately introducing nucleic acids, or electroporation, which is using a pulse of electricity to open the cell membrane. And then once they bind, we can make these cuts and remove the second DNA. So by removing that, that would be a deletion mutation. And then we could insert a new section of DNA with homologous pairs, which would be an insertion mutation. Now, there is an error because a certain percentage of cells can be misrepaired and then result in deletion mutation again, which is part of the risk of zinc fingers. However, we don't usually have range shift mutations from errors in zinc fingers because we are working with the amino acids themselves and not the individual base pairs. So, for example, here, if we go to remove these 2 proteins here from the sequence, like that, we do not affect these proteins here. Now, if we insert any section as well, as long as that is inserted in groups of 3 bases or codons themselves, then there is not a range shift mutation. Now, what's good about this is that these mutations made by zinc finger nucleases are permanent and they're heritable. And we can do that through a single transfection. And then they can also knock out genes that are not amenable to RNA interference. Now, there are some risks, and that is
konsep dia sama macam primer. Alright, konsep sama. Tapi it's different approach lah. Alright, macam the explanation in the YouTube just now, you can see that the zinc finger family works in groups. Meaning that you need to have 2 or 3 zinc finger family in order for you to cut a certain region. But you can customize it. Cuma zinc finger family ni, the editing are more into short sequences, not long. Alright, so kalau you nak edit long sequence of protein, ZFM memang agak susah untuk you dapat the mutation that you want or the deletion or even insertion that you want. Alright, so tu antara, but ZFM is the first method that not like the first, more into the first way on how scientists learn about. There's a lot of cutting mechanism inside gene in genome. Alright, so you can manipulate it as is. Okay, kalau you already research about RNAi, so RNAi is probably a natural selection category, meaning that dia ada dekat intron of the higher organism. So for RNAi, rarely it is rarely used in microorganisms like bacteria. Why? Because the RNAi are in the intron area. Alright, kalau you perasan ada exon dan intron kan? So usually kita fokus on the exon. So the intron kita assume as a non-coding sequence. So untuk non-coding sequence, biasanya we always say that, sorry, I lumputan dah come back. Tak ada kata English. Alright, so usually for non-coding sequence, it doesn't code for any protein, but it has function as a regulatory sequence. Meaning regulatory gene, meaning it help in expression of the coding sequence. Meaning it give the initiation for gene expression. So that's the function of non-coding sequence usually. Alright. So okay, that is the FM. So tak apa, later we learn more about the FM. Alright. So this is some description of the FM. So I'll try to read yang mana penting je lah. Alright. So you need to know that the FM consists of 2 components. The first one is DNA binding domain and the second one is DNA cleavage domain. So dia ada 2 domain, satu untuk bind, satu untuk cleave. Alright, and then these 2 domain are joined together to make a ZFM. So kalau orang tanya ZFM tu macam mana, dia adalah 2 kompleks. Satu ada binding domain, one consists of binding domain, the other one consists of cleavage domain. Alright, then DNA binding domain is also called zinc finger domain. So untuk binding domain kita panggil zinc finger domain. It's named because it has DNA binding zinc protein which are finger-like shape. So why is named zinc finger nucleus? Because of the shapes of it. Alright, several zinc finger protein or repeats, each repeat recognize 3 nucleotide bases. Alright, for ZFM, repeats itu only recognize 3 nucleotides. So dia recognize 3. So kalau you panjang, it's not good enough. I mean, if the sequence is long, it's very hard for the ZFM to work. Alright, because you need a lot of complex there. Alright, when you have a lot of complex, the tendency to doesn't get the origin of interest. Takut kau tu. So typically a ZFM contain about 3 to 9 zinc finger repeat, each identifying 3 bases. Alright, so DNA cleavage domain also, this one is quite important. So fork 1 only present in ZFM. Okay, untuk fork 1 ni, so the discovery of fork 1 is for ZFM family lah. DNA cleavage domain contain nucleuses like fork 1 from flavor bacteria. It recognize the DNA binding domain and cleave where it attached. Sorry, right. So this is the function of fork 1. Okay, and then each ZFM typically recognize 3 to 6 nucleotide triplets because the nucleuses to which they are attached only function as a dimer, like the explanation in the YouTube just now. Alright, pair of ZFM, Z and F are required to target any specific locus. One that recognize the sequence upstream and the other recognize the sequence downstream. Kalau you ingat balik kita punya YouTube tadi kan ada atas dengan bawah kan? So that's the meaning of that upstream and downstream. So you need to have a set. So at least depan belakang, if it's in the middle, so you need 4 set. 2 set lah for ZFM complex. Alright, in order for you to insert or delete the sequence. Okay, how ZFM work? So macam mana dia berfungsi? Alright, ZFM work in dimeric form. Maksudnya dalam berdua-berdua, dimer. Okay? Each attached at one strand of DNA along with the DNA binding domain. The DNA cleavage domain is also assigned. So, okay, it's usually not in dimeric form, but upon recognizing the binding site, it dimerizes and activates COP1 nuclear cell. So, maksudnya, in normal condition, the ZFN does not work in dimer, but whenever they recognize sequence of interest, it will form a dimer in order for it to work. Alright? ZFN is used both as for gene knockout as well as insertion. Okay. Nantilah saya pun ada punya ni. Alright, so both insertion for so this is the one I explained just now. Insert and delete the gene. Alright, and after cleavage, based on homolog directed event system of the cell, it will incorporate the EPID into the DNA. So, pembuang insertion and deletion doesn't jeopardize the other sequence. It just maintain them. Meaning that if a deletion, so delete permanently. If it insertion permanently, you will it can be inherited like the video said just now. Alright? So, usually kalau cuma tak tahu the mechanism, dah okay. Owning out pun tak boleh lah. Okay, alright. So basically kalau you tengok kat sini, so these are the how it works. ZFM pair delivered into the cell by transfection ataupun electroporation. So this is the process. So it was inserted inside the nucleus. So here is the ZFM pair recognized. ZFM pair recognized and heterodimerized around target site. So bila you dah tahu the target site, because here you create the target site punya sequence, right? Alright, so dia tahu kat mana site dia, then fork 1 will take place here. So ZFM pair makes double-stranded break and dissociate from DNA. No-repair template co-transfected with ZFM pair. 1 to 20% of cell are mispairing, resulting in gene deletion. So kalau mispairing berlaku, it will create a deletion, meaning nothing will be inserted there, just cut. Nothing will be inserted. That is mispairing lah. And then repair template co-transfected with the ZFM pair, cell contain gene integration. So if it's successful, you are able to have the insert here. So untuk ZFM it's quite straightforward lah. Dia tak se-complex. CRISPR pun almost similar like this the process. Talent a bit complex and talent and tannin I. Right? So what are the ZFM pro and cons? The pro is the first gene editing tools, so it paved the way to new horizon in genetic engineering. So bila you this one is when we're talking about pro, this is 20 years back punya pro lah. Alright. So and also enable us to learn quite a lot about genetic engineering, gene knockout and gene knocking. Kalau you perasan memang you tak perasan lah waktu tu you baby kot. You how old are you waktu 20 years ago? 3, 2, 1 years old. Wow, so young ah. 1, 2, 3. 3. Mostly tak tahu. 2. Okay, roughly. Okay, how many of you 1 year old during 20 years ago? So young. Wow, so young. 2 years ago? Oh, 2 years? Okay lah. 3 years? Oh, majority is 3 years ago lah. So, how old are you? 20 now? 23? 22 now. Yang 2 lah. Yang 23 lah. 20 years ago kan? Alright? So, basically, that's it lah. Okay. So, basically, it is the technology of 20 years ago. But it's the pioneer one. Alright? For gene editing. Okay? The con is that they have very high ratio of off-target mutation. Usually biasalah the initial one always have high chances of off-target lah. This is the first one lah. You never expect the first to be that great, alright? And then because of its zinc finger repeat recognized only 3 bases, make it error-prone, alright? So moreover, ZFN is foreign to the body. Study has been shown in animal model and causes immunogenicity. Usually, sebenarnya ZFN still be utilized. Why? Because it's cheaper. Alright, but now CRISPR also cheap. So people dah jarang. This is very rare lah ZFM now. Alright. And then we move to talent. So talent ni maksudnya dia ada bakat. No. So talent is actually it's more like 2ZFM. It's made of two parts. Satu kita panggil dia transcriptional activator like effector. That's why I call the name talent. Transcriptional activator like effector protein. And then DNA cleavage domain. So nama dia tail tu datang daripada this one lah. Alright. So DNA cleavage domain. So ada dua lah sama. Satu dia nak transcript activator. Satu kan nama dia DNA cleavage. Okay. Remember kalau katakan you tengah jawab periksa ni. Nak tahu tak ingat apa nama, tak ingat lah apa function tail, Taylor, ZFM. Always remember dekat nama dia ni sometimes ada dia punya function. In case you main breakup. Alright? Okay. Alright, so transcriptional activator like effector, tail. Tails are protein which act as a transcriptional activator. So maksudnya dia starter of the transcription process. Alright, their mode of action is the same as It is the same as them as also bind to a specific sequence in the DNA. So sama, dia ada sequence dia, alright? Then you have tail proteins present in bacteria, xanthomonas, where they bind to a promoter and switch on or off a gene. Tail when used as DNA binding protein that attach to specific single nucleotide. Okay, compare dengan ZFM, dia untuk tail dia lebih spesifik. Satu tail dia boleh attach to specific single nucleotide. So making the process more precise. Meaning that if you want to do the deletion or insertion or mutation, so it's more specific, alright? Because dia attach kat satu, ZFM dekat tiga. So the probability of the tiga tak kena is higher, alright, daripada single nucleotide. And then we have the Then we have DNA cleavage domain, just like the ZFM contain form 1 from Flavobacterium. So nama yang ni dia panggil DNA cleavage domain for the enzyme for cutting. Alright? So, ni lah yang paling panjang ni, marilah kita baca bersama. Alright? So, talent, ni nama dia. Transcription activator like effector nucleoside. For me, it's not that hard lah. Sebenarnya, very straightforward. Are similar to ZFM in that they use DNA binding motif to direct the same non-specific nucleus to cleave the genome at specific site. So function dia lebih kurang. Beza dia cuma single nucleotide, yang tu triple. Okay, triple nucleotide. And then the interaction between talent-derived DNA binding domain and their target nucleotide are less complex than those between ZF sebab dia tak works in dimer compared to ZFM. Okay, alright. And their target trinucleotide and designing talent is generally more straightforward rather than ZF. So apa beza dengan ZF kat sini lah. Alright. A key advantage of botanic is that they are not limited to mutagenesis in mouse embryonic stem cell. So disebab so mungkin pada zaman dulu, for a long time ago, people always use mouse as the tools for modification. So untuk this time, untuk talent yang ZFM ni, you can also use other organisms such as zebrafish, fruit flies, nematode, rats, livestock, and even more in monarch, and even in monarch of butterflies. So meaning that the tools can be used in a lot more organisms rather than only mice. Alright? So that's the good part, meaning that you can explore more on the genetic modification effect. Alright? So how talent work? Talent rupa dia lebih cantik sikit lah. Kalau tengok kat sini, talent ability. So first DNA binding domain to a target where each tail protein attached to a single specific nucleotide. So ni tail effector, so dia potong memang single specific nucleotide lah. Alright, so you have tail, kalau dia kiri kanan sama. Sistem dia memang macam primer, alright. All of the system is like a primer. So you have a binding sequence yang gene of target, then you cut between the middle macam tu. Alright. Alright, lepas tu okay. Alright, so the second part, the target or site of talent is composed of over 20 base nucleotide, usually 30 to 40 bases. So target dia more panjang kan? You can see it's 30 to 40 bases. Alright, so talent ni more higher punya mechanism lah, meaning that if let's say you have a long sequence of protein, so you can modify it more the longer one. Kalau short gene, you can use FM. For longer one, you can use talent. Alright? Okay, then transcription activated like effector nucleus fused with restriction enzyme for one. So another, okay, so selain daripada ZFM just now, for one was also used in talent. Alright, so this is for one type 2 restriction enzyme. What the? Okay, so for one type 2 restriction enzyme also being used in the talents. Alright, you can see here. That's major. Right? So you can see that the base pair, the target is much more longer than ZFM lah, and then the mechanism is much more straightforward than ZFM. ZFM kena ada two set kan, depan belakang, and then you can just do a normal amplification macam primer lah, alright? So kiri kanan je, alright? Kalau you perasan, lebih okay. So then Talent punya pro and con, it proves that it's simpler than ZFM, so much simpler process, and binds to only single nucleotide rather than 3 bases. Alright. Also, it's less complicated than ZFM. Its cons are they are challenging to engineer and it has problems in the delivery system. Moreover, like ZFM is also generated as immune response. Talent is time, effort, and cost domain. So talent is basically a bit higher price than ZFM. ZFM because they're small. Macam talent because it's long nucleotide, so it makes me it's a bit expensive. Tapi nowadays, these are what we talk about macam tu zaman 2-3 tahun sudah, alright? So now it's much more cheaper dah. Bukan 2-3 tahun, 10 years back lah. So now much more cheaper. Gene editing is much more cheaper now. Alright, this one. And then this is your favorite one, CRISPR. Siapa tahu CRISPR apa? Anyone know about CRISPR? Bukan that the main reason you enroll for this course? Or is it because it costs the dance? I mean, some people take this as elective. I'm not sure who takes minor elective because they want to learn CRISPR, right? They want to know more about CRISPR. There's some of your friends like that, right? So basically, CRISPR, or we call cluster regularly interspecies short palindromic repeats. Boleh keluar sendiri lah. Yalah. First identified in E. coli 1987. Tahun saya lahir. Tahun. Naturally evolved as prokaryotic adaptive. Kacau lah. Okay. Alright, so naturally evolved as prokaryotic adaptive defense mechanism. So basically CRISPR ni untuk defense mechanism. Alright, one key feature of each CRISPR occurs in the presence of an array of repetitive sequence interspaced by short stretches of non-ribosomal. Bunyi tu macam, siapa faham ayat tu? Apa? Kita dah bawa pukul 8.30. Ni kacau saya, kau apa ni? Doktor ni kacau lah tanya soalan macam ni. So basically it's actually a repetitive sequence, CRISPR. People found out that in E. coli during that time, there are repetitive sequence that are responsible for initiation of a translation process. For a transcription process of a DNA. So meaning that this sequence has some function for that particular organism. So the non-coding CRISPR array is transcribed in cleave. So remember just now when I speak about the non-coding sequence, alright? So the non-coding CRISPR, so that sequence is basically a repetitive sequence, but we do not know the function. And people, when that location was modified, then you can see a lot of changes happen to the host, to the organism. So that's why they found the CRISPR locus. They assuming that these CRISPR locus, they call it CRISPR locus. They call it another name lah during that time. That locus area must have something to do with genetic and hereditary of the bacteria. During that time, E. coli lah, alright. So then they studied the non-coding. So and that they call it as a CRISPR lah. The non-coding CRISPR array, it transcribes and cleaves within direct lipid into short cRNA containing individual spaces sequence which direct charge nucleus to the target site photospaces. So basically, this is a normal CRISPR location for E. coli. Alright, CRISPR location for E. coli. Bear in mind, this is in the first research lah. Alright, so what they do, they find this locus region as well in Streptococcus pyogenes. Alright, so mula-mula dia dapat this repetitive sequence dekat E. coli, then dia tengok the repeats in other higher organisms. Streptococcus is a bit bigger than E. coli lah. Alright, so it also have this sequences. Okay, then this is the Cas9 part. Okay, Cas9 is actually a molecular scissors. Alright? So when they know about Cas9, let's explore the Cas9. Okay? So this is actually the concept. Okay, first transcription of the pre-cRNA array and this is for Google cRNA dengan tra-cRNA. Google. Tak ingatlah apa dia punya nama full. This is from chromosome, is it? cRNA. Cepat Google. Okay, korang baca kenderaan. Trans-activating CRISPR RNA. This is for the sequence. cRNA, pre-cRNA. Okay, so this one is trans-activating CRISPR RNA, meaning that these are the RNA that are required for CRISPR analysis. This one, cRNA, I think ada soalan ni kan? CR tak jumpa. Don't found this one. Pre-cRNA. Tak dengar. Apa tadi? I dengar soalan dia jauh. But pre-cRNA. Tak apalah. It's your task lah to find out what. But these two RNA sequences are responsible in CRISPR sequences. Alright? The initial CRISPR sequences. Okay? Alright, so you can see here lah. Alright, so this is the transcriptional apa? Talent, ingat RNA itu. And then this is the other side. Alright? So this is the pre-cRNA transcription, meaning that the presence of the sequence of Cas9 here. So what happens is that maturation of cRNA occurs when the bindings of the trans to the lipid region of the So basically, this is not CRISPR process now. This is just to let you know where the location of the CRISPR and what it do inside the host, inside its own host. Alright? So third, major cRNA complex direct Cas9 to the target in which the spacer of the cRNA and the photospacer of the target is next to the photospacer adjacent. So this one is too much takut. Nanti I bagi you video lah, it'll be easier for you to see. Alright, so basically, when you have the Cas9 complex here, so you have the Cas9 complex there. Alright, so what they do, it will mature into pre-cRNA transcription. Alright, bila dia dah mature, so that is the normal Cas9, Cas9 that people use. Alright, so when they mature, you get this kind of complex where the Cas9 are inserted inside that. Alright, so in those Cas9, you have this one. So you have the target recognition. Okay, macam mana Cas9 recognize the target recognition? That's important, alright? Cas9 recognize the target recognition is by additional extension, right? Meaning that you insert additional extension that give a repetitive sequence. So Cas9, they can recognize the repetitive sequence and it can cut those sequences. Alright? So this is the general of CRISPR lah. Alright? So this is additional okay. So these are so this is types of CRISPR biogenesis. Okay. CRISPR, dia kan I don't know whether you guys know or not, CRISPR can cut banyak tempat, correct? Because it's based on the repetitive sequence. It's not single nucleotide, something like that, or you need to create a primer. So meaning that if you insert your repetitive sequence, your sequence inside the DNA sequences, Cas9 will cut it inside the genome, right? That's why you can use it in various organisms, various tools as a various tool for any gene editing, alright? So there are three types of CRISPR. This one jangan risau, nanti Dr. Kumita akan explain more. You got the type 1, type 2, type 3. So this type of CRISPR cut in different mechanism in some sort of site recognition, something like that, alright? So this one jangan risau sangat, alright? For me, for my part, I only if let's say the question for your exam or test, For me, it's that what are the difference between three methods, something like that. Not on the mechanism. You for my part. Right, mechanism, I think Dr. Kumita will ask you about the mechanism. Okay? So these are CRISPR pros and cons. Pro are that fast, cheap, and compared to other gene editing tools. Moreover, you can design easily in a way that you just insert the repetitive sequence. Alright? It cons that you also have high percentage of target mutation. So sometimes the repetitive sequence is already there in the genome. So of course you can hit the off-target mutation as well. For CRISPR, it's cheap but high off-target mutation as well. So 50-50 lah. Alright, so these are the table for you guys to have a look and compare the pros and cons for those three methods. And then you also can watch this video. I already find one video that explaining the difference between CRISPR, RNAi, and pellets. Alright? And then these are some review articles for RNA-guided genetic silencing. So whatever I give to you, so these, I think all of these below these are example of the CRISPR technology, the MN technology, and also RNAi, pellet. Okay. Alright, so these are all the papers that good for you guys to read. Okay. I think that's all. This